أهلا وسهلا بالجميع. For the sake of time خلينا نبلش. برحب فيكم باسمي وباسم بيان. راح أترك التعريف لبيان لما تبلش هي البرزنتيشن. بعرفكم شوي عن نفسي. اسمي دكتورة روان عجيلات. معي دكتوراه PhD in Clinical Pharmaceutical Sciences, Specialty Pharmacogenomics from the University of Iowa. عندي كمان Bachelor's Degree of Pharmacy from the University of Jordan. أهلا وسهلا بالجميع الفارماسيستس and non-pharmacists اللي معنا بالسيشن. راح أبلش السيشن because we do have limited time and the topic is a little bit يعني كتير كبير إننا نختصره بنص ساعة، بس راح أحاول أغطي as much as I can. So my topic today is about the role of pharmacogenomics in the drug development and clinical practice. So let's start off by defining what is pharmacogenomics. Pharmacogenomics is usually defined as the study of how genes affect a person's response to drugs. So for a typical scenario, so we have a group of people who have the same disease. They are given the same medication. Some people will benefit from the medication and they will not suffer from any toxicity. Some people will benefit, but they will have some type of toxicity. And some people will have toxicity and no benefit at all. And some people will have no toxicity, but also they will not have any benefit. So the promise of pharmacogenomics is personalized medicine, is to be able to select this group of people who will benefit from the medication and will not show any toxicity. At the same time, excluding those people who will not have any benefit from the medication, إنه ما نعطيهم any medication. And for the group who will have some benefit but also show some toxicity, we can review what can we do about it. For example, if it's dose related, we can reduce the dose. So that's the promise of pharmacogenomics. Now, when we talk about pharmacogenomics, we talk about genetic variations, which are natural differences between people. They are physiological, natural, not pathological. يعني التغييرات طبيعية بين البشر. So we have SNPs, insertion deletions, copy number variations. راح أحكي لكم عن a couple of them. So we can understand the topic. هلا my presentations a little bit starts from the beginning لأنه في عنا a lot of students. So we start from the beginning and try to get a comprehensive idea of the topic. هلا a single nucleotide polymorphism, it is the most common variation. For example, look at this point. So we have this base pair, the CG. It can be replaced by TA. So this could be a natural variation. يعني ممكن يأثر لي على البروتين، ممكن ما يأثر لي على البروتين. SNPs can also be triallelic. يعني مش بس إنه here G يتحول ل GC يتحول ل AT. ممكن إنه G to be replaced by an A or be replaced by a T. So, for example, in the multi-drug resistance gene, or as it's called, the ABCB1 gene, we have a SNP at the position 2677. G is, it is written this way, G, بعدين علامة, بعدين T to A. So, G is replaced by T or A. هلا, these SNPs are referenced and cataloged, and they are given reference numbers. Example, RS 2032582. So, what does this mean? As we know, DNA codes for amino acids. Amino acids form the protein. So this leads to differences in the amino acids and could lead to a change in the protein. So the resulting protein from this gene could be non-functional or could have a lower functional ability or a higher functional ability, or the SNP could have no effect, or it's called non-synonymous. Now, before the Human Genome Project, يعني before سنة 2000, we used to study these SNPs in a limited way. The Human Genome Project was an international project هدفه كان to sequence the human genome يعني نحصل على sequence تاع DNA تاع الهيومنز. So in 2001, the human sequence or the human genome was published in the journals of Nature and Science. The complete sequence was published in 2003, 2 years after the year 2001. The second variation, type of variation, is copy number variation, which basically means إنه الجين نفسه في عندي multiple copies of the gene. For example, the cytochrome 2D6 enzyme, it's a metabolizing enzyme. في عندي multiple copies of this gene, up to 13 functional gene copies have been observed. Some people, they don't even have a functional copy. So if a drug is metabolized by this enzyme, كتير مهم إن نعرف إيش هو الجينوتايب اللي عند الشخص حتى يعني ممكن يأثر لي على السيفتي أو الإفكاسي للميديكيشن. هلا ليش في عنا هاي ال high numbers of copies of the gene? It offers an evolutionary advantage. بيعطينا زي advantage أثناء الإيفولوشن عشان إنه جسمنا بتعرض ل xenobiotics أو foreign substances فبصير لها metabolism أحسن بهذا الإنزيم. So after the Human Genome Project صار في عندي full sequence. We have around 3 million SNPs. A lot of copy number variation, a lot of more variations. So now we have all of this data اللي ممكن إنه نستفيد منها أثناء drug development. So the genetic differences that are likely to be of most relevance to drugs are the genes that are relevant to drug pharmacokinetics اللي لهم علاقة بالabsorption, distribution, metabolism, and excretion. Also, variations that code for drug targets. So if the drug target is a receptor or if it's an enzyme and there's a variation in the enzyme, ممكن يأثر لي على response للmedication. Also, important هم الgenes اللي predisposed to toxicities such as the immune reactions. And genes that influence disease susceptibility or progression. يعني إيش الgenes اللي بتخليني more susceptible to certain diseases such as, for example, Parkinson or Alzheimer, which are in the research area right now. Or with genes that can influence progression, such as, for example, most common اللي هو الكانسر. يعني في genomics has a major role in cancer treatment. What about pharmacogenomics in drug development? I'm going to review the phases of drug development to get everyone on the same page. So the drug development process, this is in the USA. It usually has a preclinical phase. في عندي in vitro studies and animal testing. I do have the clinical phase, clinical testing, and then the application for new drug application, and then marketing of the drug. So in vitro studies, we start by identifying the target, then doing the formulation, chemical synthesis, and optimization. Then we do some animal testing to test the efficacy, selectivity, mechanism of action. And then if everything seems right, we move on to phase 1 clinical trials. Phase 1, usually 20 to 100 subjects. Usually it involves pharmacokinetics. Is it safe for use in humans? Then we have the phase 2. Is it effective? Does it work in patients, which is usually 100 to 200 patients? And then we move to phase 3, which is the large clinical trials, randomized trials, usually larger numbers, comparing to the gold standard of treatment or head-to-head comparisons. And then we have phase 4, which is the post-marketing surveillance. Now, where do drugs fail, or why do they fail in this drug development process? So this was a study that was published in Nature. It has an analysis from 4 different companies: GSK, Eli Lilly, Pfizer, and AstraZeneca. And then they cite these are the causes for drug development or failure of new treatments. So we have clinical safety, commercial reasons, efficacy, formulation, non-clinical toxicology, which is the preclinical phase, patent issues, pharmacokinetics, rationalization of company portfolio, regulatory, scientific, technical, and other. Pharmacogenomics can help us in these 4 areas: the clinical safety, efficacy, non-clinical toxicology, and pharmacokinetics or bioavailability. So the application of pharmacogenomics principles in drug development, it is a continuous process. It starts with the discovery of the drug and continues through the drug post-marketing period. يعني من لما احنا we identify target, we can look at the target, check if there's variation, look at the pathway, the genetic pathway, if there are influences on the drug target. Then we move on to phases. So pharmacogenomics is involved from the beginning to end. So we have, it's involved in the target selection and pathway analysis, for predictive toxicology, in pharmacokinetics, which are the first in human studies, selecting the responder populations, stratifying and enriching responders, and also in risk management and post-marketing surveillance after the drug is released in the market. So pharmacogenomics in preclinical studies, the most important part where pharmacogenomics can take role is in the metabolizing enzymes, because we know a lot about them. هلا لا نعرف إنه metabolizing enzyme important for a certain drug, the drug has to be at least more than 50% metabolized by this enzyme. So if more than 50% of the drug is metabolized by this enzyme, and the enzyme is polymorphic, يعني في منه variation between people, it may have an effect on safety. So it is worthwhile to study variation بهذا metabolizing enzyme. So it's important to study which genetic variation affects enzyme level and activity. We do know a lot in this area, in the area of the metabolizing enzymes. Also in preclinical studies, we can study pharmacogenomics effects on transporters. So in vitro data, which is obtained prior to phase 1, prior to preclinical studies, may represent another target. It depends on the location of the transporter. So if the transporter is in the GI, it affects the absorption, or is it at the tissue, at the target site, it may affect the binding of the drug. So pharmacogenomics can influence these transporters. Now, the ability to use pharmacogenomics to predict concentration from transporters is still in development, and I expect more research to be done in the future, and more research is needed in this area in the future. Now, moving on to clinical studies, pharmacogenomics is very important for the pharmacokinetic evaluation of the medications. Typically in all phases of pharmacokinetics: absorption, distribution, metabolism, and excretion. Again, in metabolizing enzyme and transporters. So more than 30 to 50% of all clinically prescribed drugs are either metabolized by having by phase 1 or phase 2 enzymes. And these enzymes, they do have reported SNPs. And because they have SNPs, it's worthwhile to genotype patients. So the first phase we were talking about studying in vitro, in cells or in animals. Now this phase we want to genotype patients to see the actual response in real life in clinical patients. What do these SNPs, how can they affect the drug? So if the SNP leads to a higher enzyme level, this enzyme metabolizes the drug more, so the level of the medication or the plasma level will be lower. So if the plasma level of the drug is lower, it may be below therapeutic concentration and may cause a failure in efficacy. So, and if the metabolizing enzyme is non-functional, for example, because of this SNP, the drug may accumulate, it may reach toxic level and may affect the safety. So if we can include this variation in our pharmacokinetic analysis, we can save the medication from failing these pharmacokinetic studies and selecting which, and then we move on to the next phase, is selecting which population can take this medication. So pharmacogenomics has a role in the pharmacokinetic studying of the medication. Transporters, they are really important, but we still don't know much about them because it's difficult to study transporters in vivo, which means in actual patients. And we expect and need more research in this area. Transporters, polymorphisms. So the polymorphisms or the transporters are actually substrate specific more than the metabolizing enzymes. So any polymorphism in transporters is actually worthwhile studying in the drug development process. Now for patient selection and biomarker in clinical development, we can use pharmacogenomics to define the disease, for example, or its prognosis. Let's take, for example, the breast cancer. So the breast cancer that expresses the oncogene HER2 new, they have poorer prognosis, and we have designed a specific medication that actually targets this receptor or this oncogene. We can use also pharmacogenomics to exclude patients. So we can use pharmacogenomics to include certain patients and to exclude patients. So if the patient has a risk of suffering from an allergy or a sensitivity to the medication, such as with the HLA-B star 5701 and Abcavir, so we can exclude these patients from taking the medication. يعني we choose the population اللي بياخد المedication اللي بيصيبهم يعني بيحصلوا على أحسن benefit, and they will not suffer from toxicity. So we are able to select the population using pharmacogenomics, which gives the medication a better chance for success during drug development. Also at the post-marketing phase. هلا احنا حكينا الهيومن جينوم project started or finished in 2003. So now in the last 17 years we had huge developments in technology, and now we have the sequence available. But for medications already in the market, and for medications that failed and were pulled off the market for some reason, toxicity, if we can do some genotyping, we can understand or we can try to understand the reason for this toxicity, and we can probably save these medications. So if a medication was toxic in a certain population who have a certain genotype, we can recommend that this medication should not be given to this group of people who have this certain genotype, and we can save the medication and reintroduce it into the market. Now, of course, this is only if pharmacogenomics is highly influencing of the toxicity. يعني بس إذا كان السبب هو بسبب genotype معين، بسبب نقص, for example, cytochrome 2D6 or some type of gene. So this is an overview. This was an overview of pharmacogenomics in drug development. How about in clinical practice? I'm going to go over شوي references إنه ممكن نستعملها for the application of pharmacogenomics in clinical practice. So I did a similar talk about 5 years ago. We only had like 5 or 6 medications that are pharmacogenomic recommendations. Now we have more than 300 compounds or 300 drugs with pharmacogenomic recommendations in them. We have several agencies involved, including the FDA, the European Agency, from Canada, from Switzerland, and from Japan. Several databases were set up, including the pharmacogenomics database. And I do recommend, if you're interested in this topic, to check out this database. So this is a screenshot from the pharmacogenomic website, and you can go on it. It has a lot of information. So I pulled out this information from the website, so it annotates drug labels. So if we have in the drug label, يعني بالورقة الموجودة بالmedication, في عندي some pharmacogenomic recommendations or recommendation, like the FDA requires testing for this medication or recommends testing, or testing is required before using this medication. يعني كيف بدنا نعرف? So we can go on pharmacogenomics database and look up the medications and see if the pharmacogenomic is required for these medications. This is also from the website. So drug label annotations. So we have 335 medications from the FDA, 134 medications from Europe, and so on. So if you click on this medication, Abcavir, this is what it says. Before initiating Ziagen, which is the generic name, screen for the HLA-B star 570 allele, contraindication presence of this allele. So it helps in the prescribing of the medication. Again, this is the medication that's used for the HER2 breast cancer example, Herceptin, which is only given if we have HER2 overexpressing cells. We also have guidelines, clinical guideline annotations, which is from the Clinical Pharmacogenetics Implementation Consortium. So one barrier of the implementation of pharmacogenetics is getting this information from the lab or from being technical to being clinical. So this group, they were volunteers. They started putting this information in evidence-based, peer-reviewed publication where you can go on it and see how you can apply the genetic information of this medication in practice. For example, warfarin. Warfarin is usually, we give it and we test the INR, we have the target INR ratio. In the label, we have recommended pharmacogenetic testing. So it's metabolized by the cytochrome 2C9. The target is vitamin K ORC1 gene. So let's look if the patient has a star1 allele and GG genotype, they should start on 5 to 7 milligram of medication of warfarin. But if they have the STAR3 and STAR3 genotype and the AA genotype of the target, they should be started on 0.5 milligram of medication. So it can help in the dosing of the medication, not just to give it or not. You can give them lower doses because they have lower enzymes and the drug is accumulating. There's a risk of bleeding. So we can recommend the exact doses based on the genotype. These are just examples like FDA recommends but does not require genetic testing. Here the FDA requires testing. So these change as we have more information, and these are included in the pharmacogenomic database website. Now, opponents of pharmacogenomics say we don't need to do genetic testing if we can do INR. It's really cheap. We don't need to do genetic testing. I want to share with you this graph. So in 2001, the cost for sequencing the human genome was 100 million. Now, if you look at 2019, the cost for sequencing your genome is less than $1,000. So you can get your whole genome sequence for less than $1,000. So we have the genetic tests. They are going to be available very cheaply, I expect, in the next 10 years. So now we have this huge amount of data. The problem will be in interpreting this data. And this is where bioinformatics, AI, and precision medicine and pharmacogenomics will become even more important. Change in our current practices is inevitable, and we need to prepare for it. And thank you very much.